Hello, and thank you for having me today. As Andreas mentioned, I'm Maureen Stolzer. I'm from the Duran Lab here at Carnegie Mellon University, and I'm going to talk to you today about efficient enumeration of tree topology statistics for population phylogenomics. It's quite a mouthful. So for my outline today, I'm going to have three major kind of sections. First, I'm going to talk about tree incongruence and incomplete lineage sorting, which I'll refer to as ILS. And then we're going to look at some algorithms and software we've been developing for assessing gene tree incongruence by enumerating different topologies. And finally, we're going to look at a case study of the Drosophila species. So just a brief history over time. Right in the past 50 years or so, we started sequencing genes, which led to us sequencing genomes, and then sequencing more closely and closely related species. And what we've noticed is that even when we're looking at single copy orthologs from different species, we tend to get gene tree incongruence. So when we reconstruct the history of these genes, they're not all the same. Right, so why is this occurring? And today I'm going to focus mainly on a population process known as incomplete lineage sorting. I'm going to give a brief overview for everybody. Right, so one of our assumptions when we're doing phylogenomics is we assume that lineage is coalesce at the first branch. So we can think of this going backwards in time. The time when lineages first meet going backwards happens in the first species in which they're both co-occurring, right? And in this case, if we make this assumption, the gene tree should be the same as the species tree. What happens if they don't coalesce in that first species? So what happens is, in this example, we have a much more rapid divergence of species, or if you have larger populations, these alleles may not coalesce together in the gene tree, right? And so now we're at an ancestral species where all three alleles could coalesce with one another. So by chance, we could have the genes in the most closely related species coalescing first, in which case we'd again get a gene tree that is the same as the species tree. However, right, all three are available to coalesce with one another, so we could have genes in more distantly related species coalesce first. And in this case, we have the gene in A and the gene in C coalesce first. In this case, we'd have a gene tree that differs from the species tree, not for any reason of changes in gene content, but simply population processes. And in this case, the gene tree will differ from the species tree, right? And again, we can have the other option where B and C coalesce first. So this is a fairly well-studied model, and there's a multi-species coalescence that statistically predicts the degree of congruence and incongruence. So based on a population, so between species divergences, the probability of having the gene tree that's the same as the species tree is equal to 1 minus. And each of the two incongruent gene trees have the same probability, which is 1/3 e to the minus 2. Right. And as t increases, the probability of observing this congruent tree approaches 1. So that's why as we sequence more closely related species, right, we're shortening time between speciation branches, and so we're observing more of these incongruent trees. One important observation here is that for three species, the three topologies exhibit a distribution of minority symmetry, where the congruent tree will be the most probable, and the two incongruent will have the same probabilities, which will be less than the congruent tree. Other reasons we can have trees disagreeing is from introgression, but again, we would expect that minority symmetry to be occurring with incomplete lineage sorting, but not introgression, right? So this is a signal we can take advantage of. Other reasons trees differ could be from gene duplication loss, horizontal gene transfer, or simply error. And for this, we're going to focus on trying to observe incomplete lineage sorting, and how can we pick out incomplete lineage sorting from these others. And again, we're going to focus on that observation of minority symmetry. Right, so as I just said, the distribution of tree topologies can provide information on the evolution of gene families, the population processes, and species evolution. And we can use this to do clade level analyses to look at ILS and a large number of branches of a single species tree. One of the challenges is that we have this other sources of incongruence, which we can think of as noise, and counting trees is hard in big species trees. So that brings us to our next step, algorithms and software. Why is counting trees hard? Right, so what I've been showing in a very recent divergence, we're looking at only 3 species, we have 3 topologies. This is pretty simple, right? You just build gene trees for all single copy orthologs, so we don't have to worry about gene duplication and loss. And then we can also then just count the 3 different topologies, right? And this is what has been done very often in the literature. So for example, with human, chimp, and gorilla. What if we're interested in more ancestral incomplete lineage sorting? So what if instead we were having a species tree of 4 species? So this is human, chimp, bonobo, and gorilla, right? As before, the branch where the interesting ILS is up here. So now we've got a species tree of size 4. What can we do? Well, what's typically done sometimes is we can just ignore the bonobo, right? Then we just have a species tree of size 3. Again, then we're kind of losing information, right? We could do multiple analyses. We can do one with bonobo and one with chimp, and we can compare or contrast. The problem is now we have, we've introduced non-independence between our 2 analyses, right? And in addition, if we're looking at bigger and bigger species tree, this problem then starts to explode exponentially. We can look at topologies of size 4 and get a handle of that. So just for everyone's brief overview, please trust me, these are all 15 of the different topologies of a species tree of size 4. If I color them to give you a little bit of a better handle on this, right? We see up here the 3 trees. Only these 3 topologies really correspond to the incomplete lineage sorting on here, right? All the others, for example, have chimp and bonobo not together, which would indicate incomplete lineage sorting on a different branch, or more complicated scenarios such as duplication and loss, distant introgression, or again, error. Right? And again, when we do this, the number of topologies explodes exponentially. So our observation, or what we take advantage of, is this observation of that we still get these 3 topologies if we were just considering incomplete lineage sorting on that single branch here. Right? So again, in this case, we're just looking at a randomly large tree, and if we're interested in incomplete lineage sorting on this branch, we again have the 3 trees, incongruent grouping X and Y together, and the 2 incongruent with either Y or X as the outgroup. We can take advantage of this and ask, is there regions of the gene trees that map to these topologies, right? And one of the things we want to take advantage of is we don't really care what is occurring in other areas of the species tree shown here in gray, right? We're just focusing for this single branch on these 3 areas. So is there a way that we can pull these out of the gene trees efficiently? Right, and the answer is yes. So what we can do is we can use reconciliation, which maps a gene tree into a species tree. In this case, I have a little star here because we are doing a somewhat modified version of reconciliation. And we get that mapping, which then tells us, you know, close areas of the species tree we might want to focus in on, right? So for this branch here, we say, okay, what maps to these X, Y, and Z? And then we can look at these and then count the votes for which topology it goes to. So it could be congruent, incongruent, either one, or it could be none. So in this case, right, it would be the second incongruent tree. What do I mean by none? So there are cases where we might have no votes because there's insufficient information due to loss, error, other events occurring. We could also have conflicting data. So if we have a mix of genes from Y and Z here and a mix from X and Z here, right? This doesn't really tell us a clear-cut information about one of those topologies, right? And again, duplication, transfer, integration, and error can lead to this, right? And so our method with using reconciliation and these triplets can make these distinctions between all these four votes, right? So I'm going to give you a quick case study. How are we on time?
We have about 5 minutes left.
Perfect. Hopefully, okay. Right. So with our algorithm, right, given a species tree, and we can do a phylogenomic set of gene trees, right? We do this branching order statistics for every internal branch of the species tree and for every gene tree in our set. And we do it in one go, and it just takes a few short minutes to run. And this also then includes incongruence more consistent with gene duplication and loss. And so now we're not restricted to only single copy orthologs. So I'm going to demonstrate this on a case study from Drosophila from the literature. Right. So in this case study, it was done in 2007 from Michael Eisen's lab by Pollard, right? And it's an in-depth study that found strong evidence for incomplete lineage sorting in fly genomes. And it's a great gold standard benchmark for studying ILS. And we're going to look to see how well our method reproduces these results, right? So in this case here, we've got a very large species tree here. What they were interested in this paper is Melanogaster jacoba and erecta, right? In the past, they've had many different methods for inferring species trees, and they all kind of tended to conflict with one another. And there's thought to be a very short branch here indicative of the possibility for ILS, and they were interested in, was this ILS or something else? So what they did was they built single copy ortholog trees for the 3 species plus an outgroup, and then asked which of the 3 topologies were observed. Right, so they had 9,000 some single copy gene trees, and they did observe this minority symmetry, the first grouping Melanogaster as the outgroup with 58%. And then they used a lot of additional information that was more indicative of ILS than other events occurring, and specifically looking at mitochondrial genomes and sentinel information. And they also examined systematic error. Can we reproduce these results on a multi-gene family was our question. And so in this case, we took a set of gene trees from another publication in the literature. So this is 13,000 multi-gene families. The species tree, we did our internode triplets, and then we asked on that bigger species tree, so not restricting ourselves just to these 4 species, can we recapitulate these 3 topology statistics, right? And again, these gene families had anywhere from 3 to 144 members, and this is in, I believe, 12 species. And again, we see this minority symmetry again. And if we compare, we actually get very similar numbers for the congruent topology, and we see an improved minority symmetry here, right? So great, we can recapitulate this much more noisier data set. We get the same observations out, right? So I'm going to show you this here. We did this, that was just one branch in the species tree. I'm going to show these now as little donut charts here, where the green is the congruent and the pink and yellow are the 2 incongruent frequencies. Right, we can look at another branch in this species tree. So for in this case, we have the pseudo-obscura and persimilis group together, right? And we can ask what are the statistics for that topology versus pseudo-obscura as the outgroup or persimilis as the outgroup, right? And in this case, you might expect we have a much longer species tree branch. We see much more incongruence, right? We can actually do this for the whole species tree in one go in just a few minutes, right? And we can get the statistics for everything. And what we observe is there are branches where there's almost no heterogeneity, and we do observe some of these branches with much more heterogeneity. And I'm going to focus on this example down here, right, where it appears we are deviating from minority symmetry, and we can use a chi-square test to ask, is this really deviating? And the answer here was yes, it was. We can do this for all branches. Again, there are a couple here where we do see this deviation from minority symmetry. So this is indicative of places where ILS may not be occurring and incongruence is due to some other reason. So maybe we want to focus on those branches maybe want to focus on further studies on those branches where we have lots of incongruence. And just about time, so I'm just going to thank everybody from the Duran Lab, specifically those who've worked in Notung in the past, but also Yu Ting, who is here and will be having a poster on this at the poster session today, as well as Ben Verneau, who is now at Max Planck Institute, and then Kevin Chen and Han Lai. Thank you. I don't think I have time for questions.